Hi class and welcome to Lab 6. This will be on enzymes and you're going to work through a worksheet and work through a couple simulations that will be described on the worksheet, and then you'll answer some questions in the worksheet, and then you can just submit the worksheet on Canvas. These simulations will hopefully go a lot better than the Labster lab. I'm sorry you guys had a lot of problems with that. It really makes your computer run a lot. So I gave you guys an extra week to do last week's lab if you can get it to run or get past those glitches. And if you can't get through the Labster lab glitches, just take a screenshot of how far you did get and then tell me how many points you got right out of the total, and I'll take that into account when I grade. So for this lab will be on enzymes. And I'm going to take you through a little introduction that's on your worksheet, and then I'll take you through what you'll be doing. So enzymes are important. We've talked a little bit about enzymes in lecture. They facilitate and make possible most of the chemical reactions occurring in all living systems. So they help chemical reactions go more quickly. In these exercises, you'll be examining the effects of various physical factors like temperature, pH, and concentration on the functioning of enzyme systems. The enzyme we're going to be focusing on in this lab is amylase. And amylase breaks down starch into smaller sugars, also called disaccharides. So amylase is the enzyme, and it will break down starch into smaller sugars. In humans, this starts in the mouth with salivary amylase. So already in your mouth, your saliva is starting to break down starch, but the bulk digestion of starch takes place in your small intestine by the enzyme we call pancreatic amylase, because it comes from the pancreas. And the conditions of the intestines include a pH near neutral, which is around 7, and normal body temperature. So you're first going to click on this link and watch about a 2-minute video. And then that 2-minute video should give you all the answers you need to answer these questions. If you need to use your textbook, you can look at the chapter on enzymes in your textbook to help, but this video should give you all the answers to your questions. So that video I'll show you in a second. And then activity 1 will take you to this simulation website. It's just a Google website. And like I said, it's a simulation that should run a lot better. After watching that first video, you should be able to make hypotheses that answers these 3 questions. And remember, a hypothesis is a tentative explanation or a statement of what you think will happen. So there's 3 points here, and you need to answer these 3 questions with a hypothesis or an explanation of what you think might happen. So number 1, how does changing temperature affect the rate of enzymes? Number 2, how does changing pH affect the rate of enzymes? And how does changing substrate concentration affect the rate of enzymes? I think we've covered this in lecture, but these are the 3 main physical properties that will affect the rate of enzymes. I'm not going to tell you, but a hint is, for temperature, do you think raising the temperature or lowering the temperature will make the reaction go faster? Do you think a more acidic or basic pH will make the enzyme go faster? And then do you think a more concentrated substrate will make the enzyme go faster, or less concentrated substrate make the enzyme go faster? So those are the 3 you're going to make just an educated guess on. So I'm going to take us through, I'm just going to share my screen, my Safari screen. So here's the enzyme video. It's by the Amoeba Sisters. They have a great channel that does a lot of really good science videos that are easy to understand. So this is about, I guess it's about 5 minutes long. I lied a little bit there, a little bit longer than 2 minutes. So use this first video to answer those first questions. And then this is the first, this is a johndarko.com, his Google website, and it's an enzyme simulation. And your worksheet takes you through how to run this simulation. And I don't want you to be confused, because it really takes you through exactly what to do. It tells you what to make the concentration of the initial nucleotides, proteins, lipids, or starch. So I think the first activity says to make the initial concentration of starch at 500. And you can either type in there or move the cursor to 500 concentration of starch. And then you'll add the enzyme. So for the first activity, it has you add amylase because we're working with starch. And then you can change the temperature. I think the first one, they have you run trials at 0 degrees, and then a couple trials at different temperatures, and then at different pHs. So what you're seeing is, so your substrate or what you're trying to break down, whether it's a nucleotide, protein, lipid, or starch, we're working with starch. So you'll start with a concentration of a substrate and you'll change that concentration, and then you'll add the appropriate enzyme. So amylase breaks down starch, trypsin, polymerase, pepsin, they all break down these other substrates, or not break down, but they are important for doing the chemical reaction that breaks down starch. So you'll add amylase, change the variable of the temperature and the pH, and then what you'll do is you'll run the experiment. So right now you'll see that really nothing happened in DNA, fatty acids, or amino acids because we didn't add the enzymes that helped break those down. We're focused on the disaccharides being produced. So let's change our temperature to 25, keep the pH neutral, and we'll run it again. And you can see here when we ran the experiment this time, because we increase the temperature, we get some disaccharides produced. And that means that the starch that we started with is being broken down into disaccharides because amylase, the enzyme, is helping that chemical reaction go. When we increase temperature, the increase in temperature produced more disaccharides, meaning there were more disaccharides being produced, more starch was being broken down. So the question in your worksheet asks you how many disaccharides are produced always after 20 minutes. So each trial or each run is a 20-minute trial. And to find out how many disaccharides are produced after 20 minutes, you always look for the point at the 20-minute mark, go all the way up, and then go over. And you can see how it kind of plateaus here, the graph, meaning it flattens out. But we have about 2.4 K, which just means 1,000. So about 2,400, 2,400 disaccharides were produced at 20 minutes. So you'll run this experiment several times, several trials, changing the temperature, changing the pH. We'll see if we make the pH really acidic, what happens. And they'll give you different runs with different colors. And if you want, you can have all your runs show on the same graph, or you can just reset it, which might be a little bit easier to see. Okay, so this is the first kind of simulation. I'm going to go back to the worksheet. And again, I'm going to show you here. So this is the procedure starting with the initial start concentration. Don't forget to add the enzyme amylase. And then you'll change the temperature in 4 trials to see how many disaccharides are produced after 20 minutes. And it gives you the temperature to change it to. You need to answer a couple of questions. What's happening? Basically, you need to understand that raising the temperature does something. It either increases or decreases the amount of disaccharides produced, meaning it increases or decreases the chemical reaction activity of that enzyme. Then you'll change the pH level. So again, for everything, you're always going to start with a starch concentration at 500, and we'll always be working with amylase because we're working with that enzyme. So don't worry about the other enzymes, the other 3 graphs. Just focus on the amylase, disaccharide, and starch graph. Change the pH, 7 different trials, and then tell me how many disaccharides are produced. Again, don't forget to just multiply by 1,000 because they're giving you that little K symbol, which just means 1,000. Okay, and then you'll do it again for substrate concentration. So here's where you'll change the starch concentration, and you're going to see how changing the starch concentration, the initial concentration of the substrate will affect the number of disaccharides produced. So then we get to activity 2, which takes you through temperature on enzyme function. So you're going to click on the link, start a new game, and then answer the question about what type of device you're using. This is important. If you're using a touch screen, just touch the touch screen option. And if not, just if you're on a laptop, just click no. You'll click forward until you reach the main menu. Feel free to review the information about what are enzymes and specificity if you're still a little bit unsure about what enzymes are. And then you'll click on the experiments and then keep moving forward until you get to the enzyme and temperature. And you'll follow the instructions. So this is just another simulation. It's a little bit shorter. You'll do the simulation, describe what happens, answer a couple questions, and then you'll move on to the next concept. So this activity is a little bit shorter, but I'll take you through the link again. So let me switch back to my Safari. So here's the link. You can read through the background. You can read through what are enzymes, the specificity, and then you'll go to experiments, which will take you through the experiment. It'll take you through what you need to do. You can do the quiz to test yourself. Maybe I'll take some questions for your lab quiz from there, and then it'll give you a score sheet. So this is the experiment then, and it'll take you through a couple more introductory options. You'll enter the lab, and then you'll slowly drag the slider over to see how the reaction rate of the enzyme is affected by changes in temperature. So the temperature gives you a high and low, and it shows you what exactly is happening with the substrate in that enzyme. And as you increase the temperature, you see how the rate of the reaction eventually increases to a certain point. But if the temperature gets too hot, the enzyme starts to denature. And what that means is at a too hot of a temperature, the enzyme doesn't work anymore. It loses its shape, so the substrate cannot bind to it as well. So what this is showing that to a certain extent, an increase in temperature increases the rate of the reaction. But if temperature gets to be too much, the enzyme will denature, and that means it will change or lose its shape and not be able to function anymore. You can start over with a fresh enzyme, and that's pretty much it. It takes you through enzyme and temperature, and then you answer the questions on the worksheet. If you guys have any questions, shoot me just a message in Canvas. I try to check this messages in Canvas every day. Thanks for always giving me 24 hours to respond. I hope you guys are all doing well. Thanks for your patience as we're working through this online system, and have a great day.